The introduction video helped us to understand what amino acids are made of and the primary core structure of how amino acids are arranged. This video dives deeper into the foundation of proteins by discussing amino acids in more depth. To bring us up to speed, we will show you the fundamental structure of any given amino acid. It is the fundamental structure for all amino acids will have this particular structure with different side chains. To provide an example, we will remove a side chain from an amino acid to show you the basic fundamental structure that we're discussing. This is the amino acid alanine. We need to remove alanine side chain or R group to achieve the fundamental structure of any given amino acid. We will do that now. By cleaving off alanine's R group, which is methane or CH3, the result will be the standard amino acid molecule. If we were to add a simple hydrogen to the alpha carbon as the side chain, then we would have the smallest amino acid, glycine. Adding the methane back gives us alanine again. Or if we added two methane molecules as well as another carbon and hydrogen, we would get valine. Notice how all of the additions are coming off of the alpha carbon. Once again, this is the primary structure or the foundation of all amino acids. So why the big fuss about the fundamental structure of an amino acid? Well, it is the same for all amino acids, with the only difference being the side chain, making this part of the molecule a lot easier to remember, but the way it behaves in chemical reactions is very important. The fundamental structure of amino acid, when placed in a neutral solution that is a pH of 7.0, it becomes negatively charged on one side. It's the carboxyl end, and then also positively charged on the other end, the amino group, with the end result of the molecule having no net charge. It is dipolar. There are other reasons why the fundamental structure is important, but for this video, we only want to talk about the way an amino acid behaves in a chemical reaction. When placed in a solution that is equal to the amino acid's isoelectric point, the amino acid will be primarily in a zwitter ion form. What is a zwitter ion? It is the result of an internal acid-base reaction where the carboxyl group donates a hydrogen proton to the amino group, resulting in one side being COO negative and the other NH3 positive. What is the isoelectric point? It is the point at which the pH of the solution is equal to the point at which the particular amino acid is present primarily in its zwitter ion form. Depending upon the amino acid, each has a slightly different isoelectric point. This seems like a lot to take in, but we will get to the primary takeaway in a moment. It is important to understand that the more acidic the solution, the more amino acid molecules will convert into a positive form, as the carboxyl end will pick up hydrogen protons, resulting in a cation. But the more basic the solution, the more that the amino acid will convert to a negative form as it drops a hydrogen from the amino group, resulting in an anion. The cells within our bodies have a pH around 7, which means that most of the amino acids are in their zwitter ion form, the dipolar molecule. So why is this important? Well, here's the takeaway. Free-floating amino acids within a cell can act as buffers if required due to pH changes. Also, the primary structure of proteins are based upon the linear sequence of amino acids joined together via a condensation reaction between the negatively charged carboxyl end of one amino acid and the positively charged amino group of another. R groups or side chains, with the exception of proline, are not involved in the formation of the peptide bonds. The peptide chain grows as more amino acids join the peptide chain. Ultimately, the unique arrangement of the amino acids in the peptide chain will determine the molecule's three-dimensional structure. RDStudy.com video tutorials are produced and owned by Nutrition News Network, LLC. Any copying, reuse, or redistribution, in full or in part for any purpose without the written consent of Nutrition News Network, LLC, is prohibited. All rights reserved.